So I would like to introduce Ken Burns, who will introduce our session for today. Thank you.
Thank you, Julie. It's my pleasure to introduce for his second talk in this series, Dr. Arun Srivastava. Dr. Srivastava is a professor of pediatrics, is an endowed chair, is the head of molecular therapy in the department, and has a notable track record in the area of gene therapy. In his first lecture, he talked about the basic methodology of gene therapy. Today, he's going to talk about a few cases where gene therapy has actually been used to, I don't know whether you want to use the word cure, but to treat patients with various genetic diseases with rather interesting results. So at this point, Arun, I welcome you back. Thank you for being with us again.
Sure. Thank you very much, Ken. All right. So you know where we are. This red dot shows you this beautiful city of Gainesville. Today, I'll take the opportunity to talk about gene therapy clinical success and some challenges that have come to light more recently. Yeah, so today I'd like to talk to you about gene therapy clinical success and some challenges that have come to light recently. So I showed this slide last time, the 3 vectors that have been used for gene therapy: retroviruses, adenoviruses, and antiviruses, with some mixed successes and failures because development of leukemia in most cases. And last time I was not able to cover the gene therapy of leukemia itself. And this is done by taking patients' own T cells and modifying them using a lentivirus vector to deliver what is called the chimeric antigen receptor, or CAR. And then these CAR T cells are reinfused back into patients. And this has been a real success story. This is Dr. Carl June, who pioneered this. He's at University of Pennsylvania. And Bill Ludwig was the first patient who volunteered to be enrolled in this experimental trial for CAR T cell therapy. So here's a very brief story of Bill. Bill Ludwig was diagnosed with chronic lymphocytic leukemia in 2000. So by 2010, his cancer no longer responded to standard chemotherapy. As a CAR T cell patient pioneer, he achieved complete remission that year. After his treatment, Ludwig, a retired corrections officer, traveled the country with his wife in a motorhome and celebrated milestone events, including the arrival of new grandchildren. Unfortunately, in early 2021, he died due to COVID-19 complications. So he was completely cured. The second patient who enrolled in the CAR T trial was Doug Olson. Doug Olson was diagnosed with chronic lymphocytic leukemia in 1996. By 2010, his cancer had no longer responded to standard chemotherapy. But following CAR T cell therapy, he also achieved complete remission. Olson is a former scientist. He has taken up distance running and completed 6 half marathons. He also fundraises for the Leukemia and Lymphoma Society and helps newly diagnosed patients. So last week, Dr. June declared that this is definitely a cure because these patients have now been out for more than 10 years. More heartening story is about this little girl, Emily Whitehead. At age 5, she was diagnosed with acute lymphoblastic leukemia. And 2 years later, in 2012, she was also put on, this was the first time a child had been treated with CAR T cell therapy. And I'm just going to show you images of this girl growing up year after year. This is in 2013. So she was treated in 2012. 1 year cancer-free, 2 years cancer-free, 3 years cancer-free, 4 year cancer-free, 5 year cancer-free, 6 year cancer-free, 7 year cancer-free, 8 year cancer-free, And this picture was taken 9 years after she was treated. And this is last year, so I don't have this, I guess not enough 10 years yet. So clearly this patient has completely been cured. So this is very encouraging. So I showed this slide also last month when I talked about different vectors that have been used. So this was the retrovirus that was used. Unfortunately, it's caused leukemia in some patients. Adenovirus was responsible for the first death in a gene therapy trial for in an 18-year-old patient. I mentioned Jesse Gelsinger. So today I'm going to talk to you about the most exciting virus, which is this little guy here. This is called AAV. So AAV was discovered in 1965. So from 1965 to 1990, this was not taken very seriously. Actually, Dr. Burns used to call it a biological oddity. So a lot of real virologists did not take it very seriously. They also dubbed it, AAV stands for almost a virus. They said it was not a real virus because it doesn't do anything. Of course, all that changed when Dr. Burns reported, and I showed this slide as well, in 1990, that AAV has a remarkable ability to integrate sites specifically into the human chromosome, and he mapped the integration site to the long arm of human chromosome 19, at this precise site, 19q13.3. So this is remarkable that the virus has the ability. This is the only virus that can do it, can take its DNA and integrate into a single site into the human chromosome. So Dr. Burns also says that the University of Florida is known for two things. Of course, you all know Gatorade. And then he had this foresight thinking that AAV actually will turn out to be a very useful vector for gene therapy. And the second thing, University of Florida is known for gene therapy with AAV vectors. So he says, on second thought, let's go with gene therapy with AAV vectors after everything had been reported with retro and adenoviral vectors. So clearly Dr. Burns has seen the future. So he recruited, I think among the very first faculty members from Johns Hopkins when he moved here, Dr. Nicholas Mizichka. He was assistant professor in Dr. Burns's department. And in 1984, his student, graduate student, Paul Hermonat and Dr. Mizichka reported the creation of the first recombinant AAV vector. So basically, this is what occurs naturally in the this is the wild type AAV, and basically what Paul and Nick did was simply inserted a gene, a foreign gene. In this case, it was a neomycin resistance gene. So this was the first recombinant AAV vector that was made. Of course, neomycin gene has no therapeutic value, and we'll get to that. So here's the simple advantages of AAV as a vector. It's a non-pathogenic human virus. In other words, it causes no known disease. So this is a huge advantage. And of course, site specificity of integration is a major benefit. So you could not ask for a better vector than AAV. So I'm very quickly going to show you, Dr. Burns was also instrumental in recruiting Dr. Terry Flott, also from Johns Hopkins. And Dr. Flott has the distinct honor of being the first person to inject AAV into humans. Because this was the very first clinical trial that Dr. Flott did. Dr. Flott is a pulmonologist, so he was interested in curing patients with cystic fibrosis. So he did this trial more than 25 years ago in 1996. Dr. Burns also was instrumental in recruiting Dr. Bill Houseworth. Bill Houseworth was Dr. Burns's postdoc and also at Hopkins. When he came down here, he invited Bill to join as an assistant professor or associate professor, I believe. Anyway, Bill came down here and he was clearly the the driving force behind the very first human clinical trial, successful human clinical trial that was done here at the University of Florida. And Bill and his colleagues showed that AAV vectors can cure a form of blindness in humans. So you simply inject AAV into the back of the eye in the retina, and all these patients who had been blind for 10 to 15 years regained their vision. So this was a fabulous story, made a lot of headlines. And this paper was published in 2008. So 6 patients were completely cured of their blindness. Similar trials were done by 2 other groups, one in London and one in Philadelphia, and I'll show you that in a minute. So I'm going to show you a very brief video. This was a blind patient who, you will see this is an overhead camera shot where this patient is asked to go through a small room from one end to the other, and they simply ask the question, can he navigate through this? And how many times he bumps into objects. So every time he does that, you'll see a red circle. So they're basically counting how many times he bumps in, because he's basically blind, and how many times he bumps into the objects and how long does it take for him to traverse through to the room. So here's the subject number 3 before treatment. So here's the patient, and this is the room. You can see he's simply being asked to go from this end to this end here. And these are healthcare professionals who are guiding him. So he basically cannot see. So see, he bumps into that wall. So he's just guessing where you know, because he's basically blind. So it takes him 77 seconds and 8 navigation errors. Same patient injected in the one eye, after 6 months, you'll see the difference. So he zips right through. It only takes him 14 seconds and 0 navigation error because he can see. So this was fabulous. So then the next trial was done with an 8-year-old boy. His name is Corey Haas, and this is his story.
Meet Corey Haas. He's a lot like other children's age, with one big exception. Corey was born with a rare genetic disease that left him virtually blind.
With camera, the back of the eye, the retina is the film, and in Corey's retinal degeneration, it's the back of the eye that is not working correctly. It's like the film in the camera becoming fog and not working correctly.
Being a first-time mom, I didn't know that there was actually a problem until my babysitter. So look, he's not like the other child that I have in the daycare because he's not looking at things, he's not reaching for things right in front of him.
It's very heartbreaking to see him drop something and not see it. Just keep searching for it, says he can't see it when meanwhile it's right there in front of him.
I started feeling hopeful when we actually got the diagnosis as to what was wrong with Corey's eyes. And then from there, we just hope and pray. So
Eager to improve their son's vision and prevent potential total blindness, entered Corey into a clinical trial at the University of Pennsylvania, where, under the direction of Dean Capital High, Corey underwent an experimental gene therapy procedure.
Corey had his eyes dilated, and he was placed under an operating microscope so that the instruments that deliver the therapeutic virus could be placed in the eye, and that the virus could be delivered directly to the cells that need it. The virus carried the gene which Corey is missing and allowed the defective cells to produce a protein which they did not produce before this treatment. And that resulted in restoration of the biochemical blockade in his disease, allowing him to see.
While the restoration of Corey's vision of one eye was recognized throughout the world, therapy it was felt and seen as dramatic.
We were going to the zoo 4 days after surgery. And so we're walking in to the zoo and he goes, "Sun's hurting my eyes." That's never happened.
It's been a long time coming to have some success in gene therapy. And I think we're just seeing the glimmer of the first real successes in gene therapy now.
The success of this particular gene therapy for Corey and Leber's congenital amaurosis was a real milestone in the field, that it showed that in fact you could do this type of treatment and restore vision.
It's indescribable, the emotions that I go through in seeing all of these studies take place and seeing the results. I myself have cried when I've seen Corey's. It's just amazing to me. But what is even more amazing to me is how incredible these patients are in this clinical trial. They are the real pioneers.
So this is really very exciting. So as I said, this trial was done by Jean Bennett and her colleagues at the University of Pennsylvania. And a similar, almost identical trial, just like Dr. Hauswirth's, was done by Robin Ali and his colleagues at University College in London, and they got identical results. Kathy High and Jean Bennett went on to form a company called Spark Therapeutics. And this company developed the drug, AV as a drug, called Luxturna. And this was quite successful. So Luxturna, which was made by Spark, was actually bought by the way, I should tell you, it's very expensive, $425,000 per eye. And of course, patients want to be injected in both eyes, so the total cost is $850,000 per patient. And obviously, Big Pharma saw a big money-making opportunity. So Roche, which is a Big Pharma, obviously, acquired Spark Therapeutics for $4.8 billion with a B. Barry Byrne, who is our current director of the Gene Therapy Center here at the University of Florida, was involved in a trial for a disease called aromatic amino acid decarboxylase deficiency. This trial was done in Taiwan, but the vector was actually made here at the University of Florida. And these patients are born with a missing enzyme called AADC, and this enzyme basically the lack of this enzyme renders them basically paralyzed. And here's that first child who was treated in Taiwan. She was 4.5 years. And you can see this is before treatment. She just basically just sits there. Her parents are trying to entice her to do something, but she obviously can't move. And then a single injection was given in her brain. This is the same patient a year later, 5 years and 7 months. And you'll see that she has thrived. She has gained weight, obviously, and she's now able to sit up, that you'll see. So I've seen a video. This girl is now able to run around. So this is very exciting. AV vectors have also cured another form of blindness called choroideremia. This trial was done by Robert McLaren in London. And in the meantime, a number of additional AV serotype vectors became available. So everything I've told you so far was done by AV serotype 2. And it turns out that these vectors have quite distinct tropism for given cell types. For example, AV2 and AV8 can target the liver very well. AV1, AV6, and AV7 will target skeletal muscle. AV5 will target the lung very well. AV9 targets the heart, and AV4 and AV10 target the brain. So this is very interesting. It's like you can pick and choose what you want. The only caveat here is this was all done in mouse models. So you'll see this actually becomes a problem a little bit later on. So AV1 vectors were used to treat a disease called lipoprotein lipase deficiency. This trial was done by these people in Amsterdam. And it was so successful, they actually formed a company called Unicure that developed AV1 as a drug called Glybera. Glybera was approved by the European Commission in October 2012 for the treatment of a subset of patients with lipoprotein lipase deficiency, a potentially life-threatening orphan metabolic disease. So they simply gave intramuscular injection of Glybera, and the 2 patients were cured. The problem was this drug was extremely expensive. They priced it at 1.11 million euros per injection per patient. So nobody could really afford it. And as I said, only 2 patients were treated. And subsequently, Glybera was withdrawn. But the point is that the Glybera was the most expensive drug on earth. Hemophilia B, which is a clotting disorder, was treated with AV8 vectors because AV8 vectors work extremely well, at least in the mouse liver. But based on this, this trial was done by Amit Nathwani in London and Andy Davidoff at St. Jude's Children's Hospital. And these patients made between 8 to 12% of human factor 9, which is therapeutic. They followed these patients for 3 years. And unfortunately, the levels dropped down to 5%. So clearly AV8, although works great in mice, is not the best AV serotype for human liver. And then, of course, 2 other trials were done, one by Kathy High using a modified AV8 vectors. And even though these patients made higher levels, 14 to 81% of factor 9, these patients were only followed for less than 2 years. So we don't know the long-term efficacy of these vectors. A group in Germany, Wolfgang Maisbach and Frank Lebeek, tried AV5 to treat hemophilia B. They got only about 7 to 12%. And again, they followed these patients for less than 2 years. And this is Jude Samulski's vector. This was actually a company that was called Baxalta that tried the trial with the first generation AV8 vectors. And only 1 patient made about 20% of factor 9. And but they did follow this patient for 4 years. And all other patients, and unfortunately, did not benefit from this trial. So again, the point is that AV8 was chosen because it works so well in the mouse, but clearly it is not optimal for human liver. So, and this has become more apparent recently. AV5 vectors have been also tried. This was a trial done by John Passy and his colleagues. And this was very, very efficient. These patients made between 100 to 200% factor 8. So this was quite exciting. And then another vector that's called AVLKO3 was used by Kathy High and her colleagues. And these patients made 16 to 49% of factor 8, which is clearly therapeutic. So even though these results look very promising, and you'll see in a minute, this has not turned out to be an ideal story. Before I get to that, let me just tell you one last trial that was reported. This is a disease called spinal muscular atrophy. These children are born with an enzyme that is missing called SMN. And as a result, these patients are completely paralyzed and the life expectancy is about 2 years. So single intravenous injection of AAV9 was given in these patients, and all of them recovered, and now these patients are running around. And this trial was done by Jerry Mendel and Brian Caspar. They formed a company called Avexis. Avexis was recently acquired by Big Pharma Novartis for $8.7 billion. And they have priced this at $2.125 million per injection per patient. So this drug is called Zolgensma. Zolgensma is the most expensive drug on earth today. So good news is that we now have 2 FDA-approved drugs. Luxturna, which I mentioned previously, cures a form of blindness called Leber's congenital amaurosis. FDA approval was obtained in December 2017. And Zolgensma, this is AV2. Zolgensma is AV9, and this cures spinal muscular atrophy, and FDA approval was obtained in May 2019. So far looks like from 1990, since Dr. Burns discovered site specificity of integration up until earlier this year, the new name for AAV is now, instead of almost a virus, is now an awesome vector. What is it? That's the question. Because about 20 years ago, we actually began to raise some questions about the limitations of the first generation of AAV vectors. And you'll see that because our logic was very simple. We said, AAV evolved as a virus, not as a vector for delivering therapeutic genes. So we cannot expect it to work just because it doesn't cause any disease, but it's not designed to express therapeutic genes. And as I said before, tissue tropism of AAV vectors in animal models was not readily translated to humans. So just because it works in mice, there's no guarantee it will work in humans. So anyway, these are the 3 main problems. So as I said, AAV evolved as a virus, not as a vector. And tissue tropism of animal models does not translate into humans. And as you'll see in a moment, because AAV is so safe, these companies began to inject higher and higher doses of AAV vectors into patients because low doses were not effective. And this has actually come at a very high price, as you'll see in a minute. So here's the beginning of the problem. So as I mentioned before, in the first trial that was done for hemophilia A by John Passy and his colleagues, these patients are making 100 to 200% of factor VIII. But unfortunately, after 3 years of follow-up, that number dropped down to 20 to 33%. And the latest number is, this is 5 years after treatment, that level has now come down to 8%. So this is clearly not optimal. And actually they went for FDA approval and FDA said no. They need 2 additional years of follow-up data. And my fear is this number is going to go down even further. This is Kathy Haas trial, as I showed you previously, with LKO3 vector, which is a shuffled vector. And these patients, unfortunately, 2 of them, actually several of them experienced serious adverse events. And in 2 patients, the factor VIII expression was lost. So this paper was just published in last year, in November of last year. So this is also not looking very good. And the worst fears were realized about in 2020 when the first death occurred in patients in the AAV trials, which is really shocking to all of us. So the company named Audentes Therapeutics was doing a clinical trial for X-linked myotubular myopathy with first generation AAV8 vectors. They were giving very high dose of 3 times from the 14 vector genomes per kilogram. And so 3 patients, 3 young boys died in June and August, respectively, of 2020. And a fourth patient died even at a lower dose of 1 × 10⁻14 vector genomes per kilogram, which was September of last year. So they say cause of death is still pending, but it's very obvious to everyone that the high dose clearly was playing a role. So this trial has now been put on clinical hold by the FDA. Lysogene, which is a French company, was doing a clinical trial for a disease called mucopolysaccharidosis type 3A. And this is with first generation AAV-RH10 vectors. And this vector was directly injected into the brain. And this patient also died in October of 2020. Pfizer recently reported in December of last year, this is again Jude Samulski's vector, the death of a patient at a high dose of 2 × 10⁻14 vector genomes of AAV9, and this trial has now been put on clinical hold by the FDA. So our conclusion is that the first generation of AAV vectors may not be safe, especially at high doses, because as I said, AAV evolved as a virus, not as a vector. And there's been several reported cases of serious adverse events in gene therapy trials with the first generation of AAV vectors. Solid Biosciences, which is pursuing gene therapy for Duchenne muscular dystrophy with AAV9 vectors, serious adverse events have been reported, and I won't read all that. Again, very high dose of 3 × 10⁻14 vector genomes, and this trial has been put on clinical hold as well. Pfizer, as I already mentioned, in addition to the death of a patient, several patients experienced much more serious adverse events, even though the same vector was being used, and this trial has also been put on clinical hold. Amicus Therapeutics is doing a gene therapy trial for Pompe's disease with AAV9 vectors, showed recently that this trial failed to reach the primary endpoint because, again, AAV9 works great in mice, but there is no evidence that AAV9 vectors work in humans. So this trial has been stopped. So again, the first generation of AAV vectors are also not optimal. And then people have been doing what is called DNA shuffling. They try to mix and match different serotypes and select the best one for their tissue of choice. And unfortunately, that is also not turning out to be ideal. So Adverum, which is doing a clinical gene therapy trial for diabetic macular edema, this is an eye disease with a shuffle vector called AAV7M8. And they injected a high dose of 6 × 10⁻11 vector genomes in a patient, and this patient essentially went blind. This was reported in April of last year. And they call it similarly clinically relevant events in 5 out of 12 patients. So my guess is that these 5 out of 12 patients also lost their vision, and this was reported in July of last year. Spark Therapeutics, I already mentioned, several patients have now experienced serious adverse events. And this trial has been put on clinical hold. 4DMT, which has developed a lot of these shuffle vectors. They've tried the Fabry disease with 4D1. We don't know what this is. Clearly, it is a mixed AAV vector has caused serious adverse events in some patients. This is the latest one, this February 2nd, which is last week. This is a company that was started by Mark K. They were doing gene therapy trial for methylmalonic acidemia, and this is a shuffle vector called LKO3. And serious adverse events were reported in 2 patients at a high dose of 5 × 10⁻13 vector genomes per kilogram. And this trial has also been put on clinical hold. So it is actually not really looking good for AAV vectors in general. So AAV vectors derived from DNA shuffling may also not be optimal. So what can we do about this? So this is where we, about 15 years ago, decided to develop what we are calling the next generation of AAV vectors. And I'll explain this for lack of a better word. We call it beyond this is generation X vectors, and the combination of the two is the optimized or AAV vectors. So let me just very schematically show you what I mean by next generation of vectors. So we basically made just a few changes on the capsid protein. So these are capsid modifications. And these turned out to be very critical residues that if you mutagenize these vectors no longer cause immune problems, and they're much more effective. So we reported this next generation of AAV vectors in 2008 and did a side-by-side comparison with this vector and the naturally occurring AAV capsid, and showed that these vectors are 10 times better than the first generation vectors, which means you can reduce the vector dose at least 10-fold. So this was a promising development. And then we also tried to modify the AAV genome. AAV DNA is a single-stranded DNA, not designed to express the genes, but we were able to modify the genome such that now what we are calling this the generation X vectors, which we reported in 2015, actually increases the gene expression approximately 8-fold compared to the first generation vectors. And of course, you combine the two, take this DNA, package them into this capsid here, which we are calling the capsid plus genome modified vectors. So calling the optimized or opt vectors, and reported this in 2016. And this obviously gives you an additive effect. So 10 plus 8 comes close to 24 increase. So basically this tells you that you can reduce the vector dose 20 times, which would be very useful. But more interesting observation was made by Roland Herzog, a former colleague of ours, whom we recruited from Kathy High's group, and they collaborated on this project. They took one of our AAV, next generation AAV vectors, and showed that this vector actually is less immunogenic because it does not induce a capsid-specific CD8 positive T cell response. And I was fortunate to have played a small role in this. This study was published in 2013. So not only these vectors are more efficient, they're also less immunogenic. And one of these vectors has also gone into a phase 1/2 clinical trial for gene therapy of a disease in the eye called Leber hereditary optic neuropathy, and shown clinical efficacy. This trial was done by Dr. John Guy at University of Miami, and Bill Hausworth was a collaborator. So this was very successful trial. 13 out of 14 patients regained their vision, and there was no adverse events whatsoever. And this was direct injection into the eye. So this was much simpler procedure. And Dr. Guy was ready to take this into a phase 3. Unfortunately, in early 2021, his mother, who lived in New York, got COVID-19. So he went to see her, and then he got the disease from his mother, came back to Miami, and late last year, he passed away from COVID. So this is very sad story. So the message here is that the next generation AAV vectors overcome a major limitation of the first generation of AAV vectors. So what about natural tropism of AAV vectors for humans? I think it's nice to be able to cure mouse diseases or even monkey diseases, but ultimately we want to cure human diseases. So we need to focus on vectors that only infect human cells. So back in 2009, we decided to ask this question: which is the best serotype for human liver? We know AAV8 is the gold standard for the mouse liver, but we did not know which is the best human liver tropic vector. And quite surprisingly, it turned out to be AAV3. Sorry, yeah, let me show you this. Yeah, so AAV3 was completely ignored by everyone because AAV3 does not infect any cell type in the mouse. So nobody cared. And fortunately, we were able to find that AAV3 infects human livers and only human liver cells exceedingly well. It will not infect mouse liver. So we could have completely missed it if we were completely reliant on the mouse models. And of course, all this work is published in a number of papers. And then we also asked the question, which is the best serotype for human bone marrow stem cells? And we reported in 2013 that quite surprisingly again, that this time it is actually AAV6. Now AAV6 does not infect mouse bone marrow stem cells at all. So again, we would have completely missed had we relied on the mouse model. And again, some of this work has been published. So finally, a word about the use of high doses of first generation of AAV vectors. I mentioned Unicure is using 2 × 10^13 vector genomes per kilogram. So I did a simple math. How many particles is that? 2 × 10^13 for a human body. And turns out it is 1.4 quadrillion particles per patient. So I was very curious to know how many cells are there in the human body. It turns out that somebody had already done that. Sender and colleagues estimated the number of cells in a 70 kilogram, they call it reference man, but I changed it to reference human. We only contain about 30 trillion cells. So if you simply divide 1.4 quadrillion by 30 trillion, it turns out that patients were being administered 40 times the number of AAV particles than their total number of cells in the body. This is a remarkably high dose. BioMarin, which was doing the AAV5 trial for hemophilia A, they were injecting even higher dose, 6 × 10^13 vector genomes. That translates to 4.2 quadrillion particles per patient. And that again translates to patients were being administered 140 times the number of AAV particles than the total number of cells in the body. What is quite remarkable is that human body is amazing, and so is AAV actually, that can tolerate up to 140 times the number of AAV particles than there are cells in the body. But unfortunately, when you go higher than that, which was shown by both Audentes and Pfizer, 3 × 10^14 vector genomes per kilogram, which translates to 21 quadrillion particles per patient, which then translates to 700 times the number of AAV particles than the total number of cells in the body. So clearly, human body is not designed to tolerate 700 times the number of AAV particles than there are cells in the body. And of course, we paid a heavy price for that. So here's the summary. To date, as of last night, actually, 276 phase 1, 2, 3 clinical trials have been performed with AAV vectors. First generation of AAV vectors have shown clinical efficacy in gene therapy of at least 7 human diseases: Leber congenital amaurosis, the form of blindness, hemophilia B, lipoprotein lipase deficiency, aromatic amino acid decarboxylase deficiency, choroideremia, hemophilia A, and spinal muscular atrophy. The first AAV drug, Libera, was approved in 2012, but only in Europe, but it has since been withdrawn. In 2017, FDA approved Luxturna for the gene therapy of LCA. 2019, FDA approved Zolgensma for gene therapy of SMA. And next generation of AAV vectors have shown clinical efficacy in gene therapy trial of Leber hereditary optic neuropathy. So gene therapy with the first generation of AAV vectors has experienced some setbacks, not to mention the deaths, but the use of we believe that the use of optimized serotype vectors is likely to cure a number of human diseases in the not too distant future. So I still would argue that overall gene therapy with AAV vectors is here to stay. So I just want to leave you with a few parting words. I don't think we should be using the first generation vectors to start with. So the use of the first generation of AAV vectors should be avoided because AAV did not evolve as a vector. Optimized AAV serotype vectors should be used in all future clinical trials, but I'm not a clinician, so I have no authority to say that. And I believe that we should really be working on human tropic AAV serotype vectors, and they should be further developed. And of course, we should stick to a lower AAV vector dose. So human body can tolerate 3 times the dose that there are cells in the body. So you can easily inject 100 trillion AAV particles, but beyond that, you're going to be running into problem. So I'm a strong advocate of injecting as low a dose as possible without compromising therapeutic efficacy. After the first patient death was reported, I really felt compelled to write a letter to the editor of Human Gene Therapy, which was published in July issue of 2020, in which I basically stated that it is quite a testament to the remarkable safety of even the first generation of AAV vectors that humans are easily able to tolerate roughly the same number, at least 3 times the number of AAV particles as there are number of cells in the body. And I left them with the following recommendation. We really should be using optimized AAV vectors, or opt-AAV serotype vectors are likely to be less immunogenic and more effective at lower doses, thus further increasing the safety and reducing vector production costs and ensuring translation to the clinic with higher probability of success. So I'm going to stop here, and I would be more than happy to answer any questions. So thank you so much.
Thank you, Arun.
Sure.
I don't see any hands yet. Are there any comments or questions? Pushpa, do you want to go ahead?
Yeah. I wanted to ask, why does it cost so much, the treatment with gene therapy? It obviously does not cost that much to produce the vector. Why is the cost so astronomical? Is it just big pharma that's making
Yes.
money?
That's a very good question, Dr. Kalra. Yes, so these companies have obviously invested a huge amount of money. So this is one way to recover the cost. But it is not as as simple. Preparing a clinical grade vector is expensive. So average trial costs about $3 to $5 million. So it is prohibitively expensive, but this is their way of making money.
If I may, another question.
Sure.
Why was such high doses used to start with?
So
Because the
yes.
vector, once injected, it's effective for so long. In the animal studies, it's almost
Yes.
like one shot and it's for life. Why did they use such high doses and take a risk?
Yes, very good question again. So, as I said, humans are not large mice. So yes, it works great in mice. We give low dose, and because mouse body is much smaller. So when you inject AAV into the human body, it gets diluted out. So they keep giving higher and higher doses. So that probably is the answer. I don't know if I explained it clearly, because unfortunately, all of these trials are being done by pharmaceutical companies. They pay very little attention to basic science, and they say more is better. And too much of a good thing can be bad. So I think if they were paying a little bit more attention to the basic biology of AAV, and Dr. Burns always has insisted that stick to basic science, that will provide the answers. And we were very fortunate that we were able to develop what we are calling, obviously, as you saw, the next generation, perhaps generation X, and the combination of the two, you have to pay attention to the basic science. And I think we will be able to solve these problems. But because, these companies have invested so much money, I won't bore you with all the details. I have approached some of these companies, but they are unwilling to, change, course midstream. They said, no, we have already invested time, money, and effort. We are not going to switch to. I actually had made a slide. I was very tempted to show it, but I'm not going to show it. So they don't want to see it. They don't want to hear it, because they made a decision. They're going to stick with it. And there's some ego involved. Once they think they know they have all the answers, but they will not admit that they were wrong. And this is very unfortunate. But what can I do?
Yeah. Well, thank you. That was a great lecture.
Thank you, Dr. Kala. Thank you so much.
Pat Harden, if you'd like to unmute.
Yes, this is a very non-technical question, but for the treatments that do work, the 7 that you put out,
Mhm.
and how is this at the cost really beneficial to humanity? Now, I know those diseases aren't percentage-wise, they don't rank very high in the human population, but no one could afford these
That's
except
correct.
maybe Elon Musk. And but so
Yes.
I don't, they spend all these millions, buy out for billions, and where are they going to make any profit if, you know, less than 1% of the world can afford it?
Extremely good question. Yes. So as you notice that all of these are extremely rare diseases, and FDA will not allow, FDA will only allow gene therapy of rare diseases because no other options are available. And, you can live without eyesight perhaps. So, yeah, you could argue that we don't need gene therapy for eye diseases. But what about diseases like, muscular atrophy, where the children have only 2 years life expectancy? You cannot put a price on life, right? So, if you're a parent, you want to see your child survive. So you will spend any money. And unfortunately, insurance actually covers the high cost of these treatments. But you're right, they are very rare diseases, very few children. I believe there are only 200 patients with spinal muscular atrophy. Yeah, so once you cure them, then what do you do? So clearly, we need to do two things. First of all, reduce the vector production cost and reduce the cost of the vector itself. And then, unfortunately, most of the patients are living in the developing world, and you need to make it affordable to patients, where the disease is. But these are not easy answers. There are lots of ethical questions. There are a lot of philosophical issues, and of course, we don't have all the answers. So, but you have to do something, right? So
Right.
that's what we are trying to do.
We seldom have all the answers. Thank you very much.
Thank you.
Very good lecture.
Thank you.
Go ahead, Ken.
Yeah, I would point out that most of the diseases are rare diseases that Arun's talking about. But hemophilia and certain kinds of thalassemia, et cetera, are much more common. And if you start calculating the costs of maintaining somebody with hemophilia using traditional therapy, which tends to work pretty well, but is also extremely expensive, it turns out that although a one shot at $1 million sounds horrendous, and it is, but it could be significantly less than the traditional therapy costs for a similar patient. So I think that's where they're going, or the aim. These very rare diseases are really examples of what can be done and which can't be helped by other methodology. So this essentially is a trade-off, and I think there's lots of questions about the pharmaceutical industry in the U.S. in particular, and the profit margins, which are large, I can tell you. And the question of would they come up with new drugs that they didn't make money? Well, obviously they're in business to make money, so they've got to make some money. Question is how to control the situation. When the Novartis bought the treatment for spinal muscle atrophy, which was really spectacular in terms of the results, they paid $8.7 billion to buy out this small company that had started this treatment. Well, if you start by paying $8.7 billion and you have a limited number of patients, you can imagine what you'd have to charge per patient to even break even eventually. And so it's that kind of balance which a lot of these companies are up against. And now that it's been shown that gene therapy actually works in certain cases, of course, there's tremendous pressure on these companies to be in on the chase. And so they're looking around like crazy for possible gene therapy applications. And what you need to note is that these companies always talk about the money they spend on research. But what's really much more common is that some small startup company, which is operating frequently on a shoestring or a little more than that, actually comes up with the vector. And when they finally get enough money to do a clinical trial, a few $million, and it looks good, then big pharma comes in with big bucks and wants to buy that company. And that's their basic research. And so it's hard to have tremendous amounts of sympathy for their argument.
This is very interesting, Ken, that you mentioned two diseases which are much more frequent, which is hemophilia and beta thalassemia and sickle cell disease. And fortunately, we have, as I mentioned in my talk, that we have identified two AV serotypes that transduce the liver and the hematopoietic stem cells exceedingly efficiently. And we are actually very close to the first clinical trial with the AV3 vectors for hemophilia. So if you give me an opportunity, I would be happy to go over that, one for hemophilia and one for sickle cell disease in the next couple of talks, whenever your schedule allows that.
We have plenty of opportunity for you.
Okay. And I should, in the interest of full disclosure, I think you know that already, Ken, but for everyone else, I got tired of waiting for these companies to fund our research. So we decided to start our own companies, two companies, one for hemophilia and one for sickle cell disease. So hopefully, we think, we have very good monkey data. So hopefully that will translate into humans. So I have lost all faith in mice. I don't believe mice will provide the answers. So we'll see.
I have time for a few more questions.
There's one hand I see. Dr.
Well,
Berg.
Keith Berg, go ahead.
Yes, so just a thought about this from the kind of research I used to do, which is totally unrelated, but often, and I'm asking the question here, is some of the things where you're developing therapies for these orphan diseases, these very rare diseases, is that not really also kind of learning what you can do with genes and being able to then possibly generalize it or use those things you learn in other areas? Is that true in this realm as well?
Yeah, so whatever you learn, it certainly can be applied to other diseases, right? So this is the nature of research and science in general, that whatever new information you gain can be applied to other areas. So by the way, I should mention that sickle cell disease and beta thalassemia combined constitute for the most number of patients on Earth suffer from those two. So 1 in 600 humans on Earth suffers from either sickle cell disease or beta thalassemia. So this is a huge number. Out of 7 billion human beings on Earth, close to 12 million people suffer from these two diseases. So it's rare in the US, only about 90 to 100,000 patients in the US, but in the rest of the developing world, especially in South Asia and Africa, sickle cell disease and beta thalassemia and hemophilia, those are huge numbers. And of course, every year babies are born with these diseases. So yeah, I think there is clear value in pursuing those avenues. But as short answer to your question is yes, whatever we learn from those trials, I think can certainly be applied to our advantage for treating other more complex diseases.
Thank you. And John Axe, if you'd like to unmute.
Yes. Thank you for a very nice lecture. And it
Hi,
must
John.
be
Thank you.
very satisfying for you as a pediatrician to see that young people get a new lease on life because of these treatments.
Yeah.
But I have a very basic question about AAV.
Mhm.
When it is not used to deliver a drug, or it is not toxic to humans. So do you and I have a certain number of, is it likely that you and I have a certain number of AAV viruses in our body?
So well clarify, I'm not a pediatrician, but thank you for the nice compliment, John. So yes, we about 90%, actually Dr. Burns can answer this question better than me. About 97% of the human population is already exposed to AAV, but that does not mean that we already have AAV in our bodies. What we have is antibodies to AAV. So when we get infected, we make antibodies and clear the virus. And this is what is happening in patients. When you give them low dose, patients will make antibodies and get rid of it. So that's how they keep upping the dose, hoping that it will work. But then beyond a certain point, it begins to cause adverse events.
So even though these viruses don't cause disease, your antibody system still recognizes they shouldn't be there and destroys them.
Right. So this is a perfect example. All of us are sitting here today because we have a very robust immune system. We have survived for so long because any invading pathogen will be clearly taken care of by our immune system. So human immune system is much more different, much more sophisticated than the system in mice or monkeys, right? So they can tolerate much higher levels, but our bodies won't allow that to happen. So you have to find that fine balance, where you can find a dose that will be therapeutic without causing adverse events. We believe, we are there now, but it has to be obviously tested in humans. I wish I was a pediatrician so I could be involved in the clinical trial, but I have to rely on my clinical colleagues who are doing that right now.
Okay, one other question. How many other non-disease causing viruses are there in humans? I mean, is this really a unique virus?
I can maybe able to answer that again better than I, but I believe AAV is the only virus that causes no known disease.
Really?
Is that right, Ken? All other viruses have been associated with the disease.
Yeah, let me, I think there's a little editing required here.
Okay, all right.
In fact, there are a fair number of viruses that don't cause diseases. The problem is if it doesn't cause a disease, it's hard to know that somebody has it. And it's only under unusual circumstances that you can find evidence for yet another virus. So there are lots of viruses around.
Okay.
And of course, the whole point of in my opinion, biologically of a virus is simply to perpetuate its genetic material, its nucleic acid. And if it can do that in a way that's successful without causing disease, that means it's likely to be even more successful. And AAV is an example. And the only reason we discovered AAV at all was because it turned out to be a contaminant of adenovirus preparations. And in the 60s, after Sputnik and biologists got electron microscopes, they could look at chemically purified preparations of adenovirus, and here was the second virus present, and that turned out to be AAV. So I think that it's a good example, but there are lots of other viruses, and I think that let me just say, give an example of something to ponder. It turns out that thalassemia is a very common disease in the Mediterranean region, and it's under absolute control. And how is it controlled? Because with the ability to do prenatal diagnosis, if a woman has a fetus that is positive for thalassemia, she's therapeutically aborted. So one way to keep thalassemia under control is to do therapeutic abortions. So you can balance that against the potential for treating a disease in a way that some people would find much less philosophically distressing. And I think that these are the kinds of imponderables that come up in this area, some of which were talked about by Bill Allen last week in the ethics lecture. So I think that we have all these issues to consider. It's nice to be able to do gene therapy and help some people who aren't otherwise capable of being helped. That's a big deal when that happens. The other thing is, if you go to North Florida Hospital and you go through the emergency room and spend about 24 hours while they do all these tests and find that almost nothing is wrong with you, if they had their way, they'd charge you over $30,000. And if you don't have insurance, that's what you get charged. If you have insurance, the insurance companies force them to be more reasonable in their charges. But anyway, the cost of medicine in the US is unconscionably high, no matter which angle you look at it from. And I think the pharmaceutical industry is part of this. We all realize that Medicare legally cannot negotiate drug prices with the companies. And the pharmaceutical companies really fight for that privilege. But whether or not that's good for the country, I think is a really serious question, which probably needs to be addressed. Other questions?
David, go ahead.
Again, thank you for really a very, very good talk.
Well, thank you.
But one question with regard to improving the duration of responses and perhaps also reducing the dose of the vector. So we have a number of autoimmune diseases which are not uncommon, for which patients undergo long-term immunosuppression. And in transplantation medicine, it's important to keep patients on long-term immunosuppressive therapy in order to avoid rejection. Has this been considered and or tried with these vectors?
So that's a very good question. As I said, I'm not a clinician, so I cannot really answer that. But what I can say is that there's no doubt in anyone's mind that the immune response to AAV directly correlates with the vector dose. So if you go, beyond a certain dose, you would have to contend with the immune response. So if you keep the vector dose lower, you don't have to worry about it, because AAV is very, very mildly immunogenic. Now, I don't know what happens when the patient is immune compromised or under immune suppression. Now, I should also mention that at least in hemophilia trials where they are giving these vectors systemically, intravenous injections, even for SMA where they're doing IV injections, all of these patients are put on prophylactic immune suppression, because otherwise, they cannot inject that high dose to start with. So People with autoimmune disease, I don't really have a clear answer for you because I don't know enough about what happens under those conditions. Ken, maybe you can answer that question.
No, I don't think I really can. Just to go back to a previous issue, about 90% of the population is immunopositive to AAV2. And the question of whether or not the virus goes away because of the immune system, I think is questionable. Free virus goes away, but virus is very good. Every DNA virus that infects us has the ability to establish persistent infection inside the cell, where it's protected for the most part against the immune response. And it's only if it gets activated that the immune system gets going against it again. So it's a very balanced system, and these viruses are very persistent. And so almost any DNA virus will hang around for a very long time. And you just have to think about herpes viruses of various kinds, and hepatitis B virus and hepatitis C virus. It just keeps going on like that. All right, Julie.
I think that's it. I don't see any other questions.
All right. Well, thank you so much again. Have a good day, everyone.
Thank you again.
All right, sure. Thank you, Ken. Bye-bye.
And this was the last session of this course, so we appreciate everyone being with us, and we hope to see you in the spring semester. Thank you.
Thank you.
All right.